In this video, I'm going to finish chapter number 1, cell structure, topic number 2, cells and as the basic units of living organisms. Now, in the last video, I finished the microscopy studies, and if you haven't watched that video yet, this one might be a bit confusing for you. So the first thing that you need to know is what are organelles. Now, just like organs inside a human body, organelles are organs present inside the cell. The lungs, the stomach, the heart, all of these are organs that are working inside the human body. And organelles are mitochondria, the Golgi apparatus, or the nucleus, all of which are present inside of the cell. So now that we know what's the meaning of organelles, we can move on to the different types of organelles inside of cell. Uh, First of all, I'll talk about the animal cell, and then I'll move on to the plant cell. So the first thing that you need to know about the animal cell is the cell membrane, and the cell membrane is this lining. This lining is known as the cell membrane, and basically what it does is that it allows the movement of things. So stuff that can move out and in is controlled by the cell membrane, and this cell membrane is partially permeable. And the reason for that is because of a phospholipid bilayer. For now, you don't need to know what's the meaning of phospholipid bilayer. You just need to know that it's partially permeable. This cell membrane is partially permeable, and it allows the movement of particles inside and outside of the cell. The next thing that you need to know is the nucleus, and the nucleus is this spherical shaped organelle, and what the nucleus contains is the genetic information of the cell, and which is known as DNA. Now, the nucleus is packed with a protein called histone, and histone combines together and that forms chromatin. For now, you need to know that chromatin is the protein which is present inside the nucleus. To reduce space, chromatin is small, and that means that a lot of chromatin can be packed inside this nucleus. Now, along with that, you also need to know what's the nuclear envelope. And the nuclear envelope is basically a cell membrane. It acts like a cell membrane. So this lining right here is known as the nuclear envelope. And in this nuclear envelope, there is nuclear pores. So meaning that there's holes inside of the nuclear envelope, and that allows the exit and entrance of mRNA and ribosomes. Inside this nucleus, you might see this dark circle, and in the exams there won't be a color image, so this dark circle would be a very black image, and basically this area right here is known as the nucleolus, and it has dense amounts of the protein chromatin, which I said earlier. So all of the protein, most of the chromatin protein is present right here, and along with that it also contains DNA. That's why it has a different color. It's more dense, so the color is more dense. So in the exams that will be shown as a black spot inside the nucleus. Let's talk about mitochondria. So mitochondria is this oval-shaped organelle which has these flaps inside of it, and in the exam this area, this brown area, would be very black. So this flaps, the lining of these flaps are called cristae, and the fluid that is present inside this cristae is known as matrix. Now, what does matrix include? Matrix has enzymes for the aerobic respiration that mitochondria does. Now, the main role of mitochondria is it is the powerhouse of the cell. So mitochondria is the place for aerobic respiration, and it gives energy to all the organelles that are present inside the cell. Ribosomes are these really small organelles. So all of these dots are ribosomes, and they are also found on the rough endoplasmic reticulum. So this right here is the rough endoplasmic reticulum, and all these dots represent ribosomes. Now, the function of the ribosomes is to translate genetic material into protein. So, genetic material found inside the nucleus, which is the DNA, is converted into mRNA, and this mRNA is moved out of the nucleus through the nuclear pores. So this pore right here will move out the mRNA, and this mRNA will be converted into protein with the help of ribosomes. There are two types of ribosomes, the 70S and the 80S. The 70S ribosome is made up of two subunits. So this 70S ribosome is made up of two subunits, the 50S and the 30S. The second type of ribosome is known as the 80S. The 80S ribosomes are found in eukaryotic cells only. Later in this video we will learn what eukaryotic and prokaryotic cells are, but for now just know that 80S ribosomes are only found in the eukaryotic cells. Inside all the cytoplasm, the small dots are 80S ribosomes. Now, the 70S ribosomes are found in two areas. So, the 70S ribosomes are found inside the mitochondria, and it's also found inside chloroplast, while all these dots are 80S. The rough endoplasmic reticulum is this tail-like organelle found near the nucleus, and the reason it's called rough is because it has these ribosomes on it. So the ribosomes make protein, and the function of the rough endoplasmic reticulum, or RER for short, is they process and they fold these proteins, and right beside the rough endoplasmic reticulum is the smooth endoplasmic reticulum. And the reason it's smooth is because it has no ribosomes. You can see that it's really smooth and it does not have any ribosomes on it. So what the smooth endoplasmic reticulum does is it's a factory for steroids, lipids, and hormones. The Golgi apparatus is similar in shape with the smooth endoplasmic reticulum, and the Golgi apparatus, or also known as the Golgi body, is a factory for proteins. What that means is it processes and modifies proteins into two things. The first thing is these small sacs known as Golgi vesicles, and the Golgi vesicles act as a storage. And the second thing that the Golgi apparatus produces is the lysosome. Lysosome acts as a digestive system for the entire cell. It contains digestive enzymes. Digestive enzymes are used to break down any old or unwanted organelles. For example, this organelle is unwanted, so it would be digested by this lysosome right here. The microtubules are these stick-like shaped organelles. All of these are microtubules, and they give the cell the shape that it has. So microtubules act as a skeleton for the cell, just like a human body has a skeleton and it has bones. So the bones of the cell is the microtubule right here. Now, microtubules are made up of tubulin, and there's two types of tubulins, tubulin alpha and tubulin beta. So tubulin alpha and tubulin beta. So this alpha and beta combines to form a dimer, and this dimer combines together, this dimer right here combines together to form this cylinder, and there's 13 dimers to form this cylinder right here. So 13 dimers combine together to form this cylinder. This cylinder is made up of 13 profilament dimers around a hollow core. The last thing that you need to know about animal cells is the centriole, and the centriole is made up of 9 triplets of microtubules. So this right here is a microtubule, this right here is a microtubule, this right here is a microtubule. Now these three combine together to form a triplet, and 9 microtubule triplets, so 1, 2, 3, 4, 5, 6, 7, 8, 9 microtubule triplets combine together to form a centriole. And for now you only need to know that centrioles are absent in plant cells and that centrioles are involved in cell division. The next cell that you need to know of is the plant cell, and plant cell is very similar to animal cells except that it has a few more organelles and a few less organelles compared to animal cells. The first thing that a plant cell has, which an animal cell does not have, is this cell wall right here. This green area right here is known as the cell wall, and the cell wall is a structural layer right outside the cell membrane. So it's not inside the cell membrane, it's outside the cell membrane, and it provides two things for the plant cell. It provides protection from outside the cell, and the second thing that it provides is support for the cell and protects the cell from collapsing, and it gives the shape the plant cell needs. The second thing that the animal cells does not have is known as the plasmodesmata. It's not found in this diagram, but the plasmodesmata is basically a bridge which connects two organelles and enables them to transport and connect. So they can transport things and they can communicate via the plasmodesmata. The last thing which the plant cell has, which animal cells do not have, is this large vacuole right here, and this large sac is only found in plant cells, and this line right here, this membrane, this vacuole has a membrane of its own which is known as the tonoplast. So the vacuole provides two things. It provides storage and it provides the structural support for the cell wall. ATP, also known as adenosine triphosphate, is an energy source which is found in the mitochondria. Now, more specifically, it's produced in these flaps of these mitochondria, and ATP can react with water to form large amounts of energy and another side product. For now, you only need to know that ATP is an energy carrier, and when it reacts with water, it produces a lot of energy. This reaction is known as hydrolysis. The next thing you need to know is comparing animal cells to plant cells. So animal cells have centrioles and lysosomes, which plant cells do not have. And plant cells have a cell wall, chloroplast, plasmodesmata, and a large vacuole, whereas animal cells do not have those organelles. Now, animal cells do have a vacuole. They have multiple small vacuoles, but they do not have a large one big vacuole, which is present in plant cells. You also need to know what eukaryotic and prokaryotic cells are. So eukaryotic cells are animal cells and plant. You also need to know what eukaryotic and prokaryotic cells are. Eukaryotic cells are animal cells and plant cells. So basically any cell which has a nucleus and is bounded by a nuclear envelope is known as eukaryotic, while a prokaryotic cell does not contain a nucleus bounded by a nuclear envelope. So eukaryotic cells are very big because they have a lot of organelles, while prokaryotic cells are usually very small because it does not contain a lot of organelles. And eukaryotic cells have linear DNA with histones, whereas prokaryotic cells have circular DNA. And eukaryotic cells have membrane-bound organelles, but prokaryotic cells does not have membrane-bound organelles. Eukaryotic cells have big ribosomes, while prokaryotic cells have very small ribosomes. You also need to know what's common between eukaryotic and prokaryotic. So both eukaryotic and prokaryotic contains a flagella, a plasma membrane, cell division, cytoplasm, and ribosomes. Last thing that you need to know for chapter number 1 is viruses. Now, viruses are non-living things. The reason for that is viruses can only reproduce within a host. So if there is no host, the virus cannot do anything else than exist. So the virus has this capsid protein code. This code right here is known as the capsid, and it also contains protein molecules attached to the capsid, and this has either a DNA or a RNA genetic code which can be used to multiply if the virus gets into a host. And with this we have completed chapter 1, cell structure.